So here is another image showing how proteins can differ. You can have all of these different ways, different categories of proteins. So actin and myosin are going to be your contractile, immunological, your antibodies, storage like ferritin that stores iron to help your red blood cells carry your oxygen to the rest of your body from your lungs. And then you have structural collagen that's in your skin. And then you have all these other ones. Okay, protein structure. What you got to understand is that after proteins get made, translated, they're going to be able to fold into different sections: primary, secondary, tertiary, quaternary. So these are just degree of complexity. Primary is a linear sequence, secondary is a shape, tertiary is when they actually fold, and quaternary is when they actually interact. So we see quaternary when they actually perform their function in, like, enzymes, for example. So these are actually going to be your tertiary structure. And this one has a quaternary structure. Okay, so primary structure is going to be like a string like this. Here's a video to help understand that those the different ways that the protein can fold. Okay, so here's another example in that what happens if there's a mutation. Okay, so mutation is when something goes wrong and the code got changed somehow, right? So a substitution for one amino acid, you can see how this one, glutamine, becomes valine, and this can cause a change in the way the protein folds, becomes sickle cell anemia. And here you go. So here is a primary structure, secondary structure, then you have a tertiary structure where they come together, and then a quaternary structure. Okay, protein is a functional molecule built from one or more polypeptides. So you can have multiple subunits. So each of these is one polypeptide, and a protein, a functional protein, would be one that has can have multiple subunits in it. Okay, here is another view of how you can categorize proteins. Some can be globular, some can be fibrous. Okay, so bovine insulin, bovine means from cows, is a small globular protein because they kind of are stuck together in a glob. And then collagen here is an example of a fibrous protein. Globular protein are easily soluble in water usually, and they usually have these type of functions. So like hemoglobin, for example, that could then hold oxygen in the middle there of each of those subunits. Each of your hemoglobin can hold up to 4 oxygen. Fibrous protein are going to be usually structural or contractile. They're going to be water insoluble, they're going to be tough. So those are the ones like collagen or actin and myosin that are found in your muscle cells. Okay, now we're going to move on to chemical reactions. So in this section, we're really going to see how it's a review from 8th grade science, basically, how reactants become product. So reactants are going to be your molecules that go in to the reaction. Products are the end product. Okay, so reactants are the starting, products are the end. Each circle is one atom. The letter tells you what atom it is. So this is hydrogen, this is oxygen. And the line here is a covalent bond. So this with 2 lines is a double bond, meaning 2 covalent bonds together. And in technical detail, what this means is that these two are sharing 4 electrons, whereas these hydrogens are only sharing 2 electrons. Each bond, let me write that down, each covalent bond equals 2 electrons shared. Okay? All right, so a little bit more about reactions. So anabolic reaction is when you're going to be making something. So it's usually going to be when you're going from smaller molecules, smaller reactants with very low energy level to bigger molecules that have high, that are storing more energy. We call that endergonic reaction, and this is non-spontaneous. You're going to have to put energy into it. Whereas catabolic reaction are going to be a result of decomposition, breaking down, and it produces energy. Okay, so one thing that we're going to come back to over and over again will be the difference between like photosynthesis versus cellular respiration. Photosynthesis requires an input of light energy to go from small molecules like carbon dioxide and water into making your glucose. And then in opposite is cellular respiration. You're going to break down that glucose into carbon dioxide and water. Okay? You have here, you see this hump, right? And another hump. And this one is what we call the activation energy. All right, so here is another view of the reaction. So you got to know that there is a hump sometimes. So in this other, what happens is that enzymes, which is a type of protein, can lower this activation energy to make it go faster, right? Um, in when we talk about, we're going to talk about all of these things in class, but I want you to be able to answer these questions with reactions that we talk about. All right, so enzymes. So we're leading, basically this whole section's been leading up to this. Enzymes are biological catalysts. They speed up reactions and they regulate cell processes. Enzymes have a specific region where they're going to bind to the active site. Enzymes are very specific, right? Meaning that you have an enzyme for each reactant. Lactase will only work on lactose. Okay, so things like that. Okay, what do enzymes do? They are proteins, they speed up reaction, they lower activation energy, usually reversible, not always. They're going to be used up. They're not going to be used up, sorry. They're not going to be used up in a reaction because they're recyclable. Okay, here's an active site, more information about that. It's a substrate, so let's say this is ribonuclease S that break up RNA. So this would be the RNA molecule that comes in and it breaks apart the nucleotide there. Active site is where it's going to bind to, it's the pocket where the substrate's going to sit. Okay, so here are steps in enzyme activity. We call this induce fit because the enzyme sometimes will open up to allow for that active site to be exposed. When the substrate molecules go in, the enzyme will change shape to allow for those molecules to fit together in such a way to break bonds or form bonds or whatever, and then releases it. Denaturization, we talked a little bit about this back when we did when we talked about beets, right? The beetroots and how increasing the temperature of the beetroots will lead to more leaky cells because it's you're basically denaturing the protein channel's shape so that the gate becomes non-functional, right? The protein channel becomes broken down and it can't do its job. So this is what happens when it's denatured. Usually the structure is pulled apart so that by extreme temperature or pH, and then the protein unfolds and no longer can do its job. The effect on pH on enzyme is that there, especially in our bodies, there are specific pH that will work optimally. And that varies from enzyme to enzyme. The neutral pH that we are going to be concerned about here is 7. Anything lower than that is acid. Higher than that will be alkaline or basic. Notice that pepsin, which is found in your stomach, work at a more acidic level because it's found in a more acidic environment, which is like your stomach, right? Then you have trypsin and urease that will work at more neutral pHs. Here is temperature. So there is going to be usually an optimal temperature for what enzymes function. Because our body temperature is going to be about normal body temperature is 37 degrees, the optimal temperature for most enzyme in our body will be 37. Sorry, did I say that wrong? I meant to say this is about 37 degrees right here. Okay, so that's the optimal temperature there. That's too cold. That is going to make it unfold. Okay, then understand that the enzyme concentration can also result in how well the reaction goes. If you have more enzyme, usually that will lead to a faster reaction, but not always. Okay. Substrate concentration, it can plateau out. Okay. And then we're going to do this catalase activity in class. So in this lab, and that should be a D, you're going to discover the optimal condition of when catalases works. Catalase is an enzyme that will work on hydrogen peroxide to break it down into water and oxygen. And then that's it for this unit. Okay, there's a lot.